Hello everyone. Today we are going to discuss about types of gene mutation. Before going to this topic, type of gene mutation, we have to know that what is the meaning of mutation. Generally, mutation refers to any type of change. Okay. When in biological science, when any type of changes occurs in nucleotide sequence that affect the physical nature of the organism. Okay. So when nucleotide base sequences are alters or changes their position or their number, then mutation occurs. So what is mutation? Mutation is the change, any type of change in nucleotide sequence or any type of change, any type of tiny type of change or alteration in genetic basis that brings physical change in an organism. Physical change in an organism. Okay, this is termed generally the mutation. Our topic is gene mutation. What is gene mutation? Gene mutation is the change in genetic form. Mutation refers to change. Gene mutation refers to change in genetic form. That is change in the nucleotide base sequences. Okay, gene mutation refers to change in nucleotide base sequences either in the number of a base sequence or in the position of the base sequence. We will going to study that is called the change in a nucleotide base sequence called gene mutation. Okay, this is the thing, gene mutation. Then there are types. What is the types of a gene mutation? There are generally two different types of gene mutations are present. Two different type of gene mutation. One is point gene mutation. Another is Frameshift gene mutation. Frameshift gene mutation. First of all, we will going to discuss about what is point gene mutation. Point mutation. See, point refers to dot, a particular area. Okay, point refers to dot, that means a particular area. So point mutation refers to when mutation or change occurs to a single nucleotide base sequence or a one nucleotide base sequence when undergo change, then that is called point mutation. Okay, point refers to a particular point, a particular area or a particular thing. When a single nucleotide base sequence or a one nucleotide base sequence undergo change in a particular gene, then that is called point mutation. Point mutation is defined as change in a single nucleotide base sequence. Defined as change in a single nucleotide base sequence. Okay, point mutation, change in a single nucleotide base sequence. That means if a particular area, a particular nucleotide base sequence is alters or changes, that means point mutation. That is called point mutation. This point mutation is three types. One is silent point mutation or same point mutation. Another is missense. First, second is missense, S-E-N-S, missense point mutation. Another is nonsense point mutation. First, we will see what is silent or same point mutation. Silent point mutation. Silent refers to when we are doing something but anyone does not know. Okay, silent refers to that. So silent point mutation refers to when a change in nucleotide base sequence occurs. Change in nucleotide base sequence occurs, but that does not bring any change in the function. What is the main function of the gene? Main function of the gene is to synthesis polypeptide chain. That means protein. Protein synthesis is the main function of the gene. Then, if change in a single nucleotide base sequence occurs, but that does not bring any change in the function and does not bring any change in the synthesis of polypeptide chain, then that is called silent mutation. Mutation occurs, but the effect of that mutation does not occur. That is called the silent or same mutation. Same mutation occurs defined as change in nucleotide base sequence, change in a nucleotide base sequence that brings no effect on synthesis of polypeptide chain or protein. Okay. When mutation occurs, but the effect of that mutation does not occur, then that is called the silent mutation. See one example. Suppose we are talking about example. Suppose the DNA sequence. Suppose the sequence of a DNA is TTC, thymine cytosine. Suppose the DNA sequence is TTC, thymine cytosine. Then when mRNA is synthesis from this DNA, then its sequence will be AG. Okay, because we know that A11T, C11G, opposite base pair of the thymine is adenine. Thymine cytosine. If the base sequence in DNA is thymine cytosine, then the mRNA synthesis from this DNA by transcription process will have the sequence AAG, adenine guanine. Then this AAG codon synthesis for a amino acid in polypeptide chain, that is called lysine. Okay, so this is an example, lysine. AAG synthesis lysine. This is the normal condition. If this normal sequence undergo mutation in a single place, in a single nucleotide, suppose this cytosine is changed, mutation occurs, T, then suppose another thymine is added, T. Suppose in place of C, another thymine is added. Then change in nucleotide base sequence of one place occurs. This is called point mutation. Then if this mutation occurs to the DNA sequence, then the mRNA synthesis from this sequence will be AAA, adenine, and this AAA codon also synthesizes the same amino acid in a protein synthesis, that is called lysine. Okay, so in this case mutation occurs, but effect of mutation does not occur, because that's why it is called silent or point mutation. Mutation occurs in place of cytosine, thymine is added, but the effect of that mutation does not occur. The same protein synthesis, same amino acid synthesis occurs in the polypeptide chain synthesis process. Lysine is synthesized by AAG in the normal condition. After the mutation also AAA codon also synthesizes the same amino acid in the polypeptide chain synthesis. So this is called the silent point mutation, where mutation occurs to a single nucleotide base pair, but its effect does not occur. This is called silent point mutation. Then the next thing is missense point mutation. Secondly, missense point mutation. It is the opposite of silent point mutation. In silent point mutation, mutation occurs, but its effect does not occur. But when mutation to a single nucleotide base pair occurs and that bring change in the function, then that is called missense point mutation. Okay, change in one nucleotide, change in one nucleotide brings change in function, change in function or alters function of a codon, function of a gene, then that is called missense point mutation. When mutation occurs and the effect of that mutation also occurs, then that is called the missense point mutation. See the same example here. If the DNA have the sequence, if DNA have the sequence TTC, thymine cytosine, if when mRNA is synthesis from this DNA by transcription process, its sequence will be A G adenine guanine. This is the normal condition, and this synthesis lysine. When mutation occurs, suppose in this thymine place T C C codon, this in place of thymine suppose cytosine is added, then change in the nucleotide base sequence occurs at one point, at one nucleotide base sequence. So when one nucleotide base sequence undergo mutation, that is called point mutation. This brings when mRNA is synthesis from this mutant DNA, this have the sequence adenine, C for guanine, AGG. This AGG codon does not synthesize the same lysis amino acid. This synthesis another amino acid. So the mutation when brings alter or change in the function, then this is called point mutation. AGG synthesis for another amino acid arginine. Okay, when mutation to one point brings change in the function, change in the synthesis of polynucleotide chain, then that is called missense point mutation. Okay, this is the case missense point mutation. Then the next thing is nonsense point mutation. Nonsense point mutation. Okay, what is nonsense point mutation? When mutation to one nucleotide base sequence brings a nonsense codon. Nonsense codon refers to stop codon. Okay, that is UAA, UAG, UG. Okay. When change in one nucleotide base brings nonsense codon or stop codon. That is stop codon. There are three stop codons: UAA, UAG, UGA. Okay. When mutation to one nucleotide base sequence occurs and that brings stop codon or nonsense codon, then that condition is called a nonsense point mutation. See one example. This is the normal sequence of DNA. When mutation occurs, suppose in this place, mutation occurs. In place of thymine and one adenine is paired. ATC. In place of thymine, adenine is added. Then when mRNA synthesis in transcription process, then from A, uracil is paired. Uracil, because in mRNA in place of thymine, uracil is present. A double bond U. The opposite of A is uracil. U, T opposite A, C opposite G, U, A, G. So when mutation brings a stop codon, this UAA is a stop codon. There is no anticodon for the UAG codon. That's why it is the stop codon, regarded as stop codon. So when mutation to one point, one nucleotide base sequence brings a stop codon, then that is called the nonsense point mutation. UAA. It is a type of stop codon. This is an example. Okay. So this is the thing nonsense point mutation. When change in one nucleotide base sequence occurs and that brings a nonsense codon or stop codon, then that is called the nonsense point mutation. Okay, this is the thing about point mutation. Point mutation refers to change in a single nucleotide base sequence, that is called point mutation. There are three types of point mutation we discussed: silent point mutation, then missense point mutation, and nonsense point mutation. Then another type of point mutation is frame shift point mutation. The next thing is frame shift point mutation. Frame shift point mutation. Okay, frame shift point mutation. What is frame shift point mutation? It is the opposite of point mutation. Frame shift gene mutation. Sorry, frame shift gene mutation. Frame shift gene mutation refers to when change in several nucleotide base sequence occurs. That means in point gene mutation, changes in a single nucleotide base sequence occurs. But in frame shift mutation, change in several nucleotide base sequence occurs, and that brings the change in the reading frame or reading sequence of the DNA of the gene. Then that is called frame shift point mutation. Mark here. Frame shift point gene mutation is defined as when change in several nucleotide base sequence occurs. Shortcut, I am writing in shortcut. Change in several nucleotide base sequence that brings change in a reading frame. Change in a reading frame of a gene. Then that is called. Then it is called frame shift gene mutation. See here, when several nucleotide base sequence occurs, this occurs due to addition of extra nucleotide base sequences or deletion of the previously existing nucleotide base sequence. See, addition of new or extra also you may say, addition of new nucleotide base sequences or deletion of previously existing nucleotide base sequence when brings change in reading frame structure or original combination of the codons, original combination of gene, then that condition is called frame shift mutation. And this frame shift mutation occurs by two processes, or we may say two types. One is addition, another is deletion. Okay, see an example. First we are going to discuss about addition frame shift mutation. This is very important for short question. What is frame shift mutation? See, by an example we will get understood better. Addition of point mutation. Suppose the DNA sequence. Suppose the original DNA sequence is, okay, forget the DNA sequence, directly come to the mRNA sequence. If mRNA sequence is like this, suppose this is the mRNA 3' and 5' end. mRNA consists of several codons. Suppose this is the first codon. Suppose it is AUG. Then second codon, suppose UGG, UGG. Okay, UGG also no doubt it is a codon. CUG, ACG, CGG, CGC. Suppose this is the sequence of the codon in a mRNA. When I am describing addition frame shift mutation, when extra nucleotide base sequences are added to a particular area, suppose CG, two nucleotide base sequence added in this area, then its structure will be changed. AUG, then third CUG. Its reading frame will be changed now. See, another ACG. Then when CG is added, then it will be regarded as CGC. See here, the original combination is CGG. When C and G, cytosine guanine, extra cytosine guanine added in place. This is the extra cytosine guanine. These are added. Then the reading frame is changed from CGG to CGC. Then this GG is changed into GGC. Then this GC. Then next nucleotide base sequence. So the reading frame is changed and it alters the function. The amino acid which is synthesized by CGG may not be synthesized by CGC. In this condition it is synthesized, but if I say AG, A and G if added, then this is regarded as AGC. The amino acid which is synthesized by CGG is not synthesized by AGC. So this type of mutation, when extra nucleotide base sequences, more than one extra nucleotide base sequences are added to the codon, and that brings the change in the reading frame, that is called the frame shift point mutation. Frame shift gene mutation. Okay. In this condition, two extra nucleotide base sequences are added, so that the reading frame of the mRNA is changes, and it brings the change in the protein synthesis process. That's why it is called frame shift gene mutation. It is a type when addition of gene mutation occurs. If deletion of gene occurs, deletion of gene nucleotide base sequence occurs. See, example. If this CG are deleted, then what is the reading sequence? AUG, CUG, AUG, CUG, ACG, ACG. Then here C and G are deleted. That means the reading frame will be changed into GCG, GCG. Then C. Then next further, if there is another is there, AGC. Then it will be read as CAG, CAG. Okay, like this. So when deletion of the nucleotide base sequences occurs or omission of the nucleotide base sequences occurs, and that brings change in the reading frame, then that is called deletion type of frame shift mutation. Deletion type. Addition, this is deletion type of frame shift mutation. When nucleotide base sequences are deleted, more than one nucleotide base sequences are deleted, and that brings change in the reading frame. This CGG, the amino acid which is synthesized by or which is decoded by CGG, is not decoded by GCG. See here, CG is changed. That's why it is read as GCG. GCG. Okay. So this is the thing of frame shift mutation. Frame shift gene mutation. When nucleotide base sequences, more than one nucleotide base sequences are deleted or added, and that brings a change in the reading sequences of the gene, then that is called frame shift gene mutation. This is the condition, this is the things discussed about types of a gene mutation. There are two types of gene mutation. One is point gene mutation, another is frame shift gene mutation. In point gene mutation, only a single nucleotide base sequence undergo change. This is that point mutation is three types. One is your silent mutation, another is missense mutation, another is your nonsense mutation. In silent mutation, change in one nucleotide base sequence occurs, but that does not bring any change in the function. But in missense point mutation, change in the function occurs. Change in a single nucleotide brings change in the function. And in nonsense point mutation, a change in single nucleotide base sequence brings a nonsense codon or stop codon. Then the thing frame shift gene mutation, when more than one nucleotide base sequence undergoes change, that may be addition or that may be deletion. addition or deletion to more than one nucleotide base sequence occurs, and that brings the change in the function, that brings change in the reading sequence of the gene, then that is called your frame shift gene mutation. This frame shift gene mutation is occurs due to addition of the more than one or several nucleotides or deletion of several nucleotides. This is the things of gene mutation. Okay. Another thing you may say, another thing you may mark, point mutation is occurs due to substitution of nucleotide base sequence. Nucleotide base sequence substitution. We see that TTC, if in place of thymine adenine is added, then mRNA synthesis is UAG. This is the substitution. In place of thymine adenine is added. In place of thymine adenine is added. This is the substitution case. Point mutation occurs due to substitution. Frame shift mutation occurs due to deletion or addition. Deletion or addition. Extra nucleotide base sequence added or the existing nucleotide base sequences are removed. That is called the deletion or addition. Frame shift mutation occurs due to deletion or addition. Point shift mutation occurs due to substitution. Okay, this is the thing. So this is the things we discussed about types of gene mutations. Then another thing we are going to discuss about split gene. Split gene. What is the condition split gene? See, in prokaryotic gene, the prokaryotic gene is continuous. That means the functional part of the gene of prokaryote is consist of only functional part. But eukaryotic gene consists of functional part and non-functional part. Okay, eukaryotic gene consists of functional part and non-functional part. See, suppose this is the eukaryotic gene. Suppose this is the eukaryotic gene. This eukaryotic gene consists of functional part and large generally non-functional part. This is the non-functional part. Again this is the functional part. Again this is the non-functional part. This is functional part, non-functional part, functional part, non-functional part. This is the eukaryotic gene. Eukaryotic gene consists of functional part in which your protein synthesis, this orders protein synthesis, polypeptide chain formation occurs through in this place, and non-functional part undergo non-functional part does not have any function. That's why it is non-functional part. The functional part of eukaryotic gene is called exon, 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 and the non-functional part of eukaryotic gene is called intron, INTRON. Okay, so split gene refers to when a gene consists of functional and non-functional part. That is called split gene. The gene which consists of functional and non-functional part alternate with each other or present one after another. One functional part, one non-functional part, another functional part, another non-functional. When functional part and non-functional part of a gene present one after another or alternatively with each other, then that is called the split gene. Split refers to separation. Here the functional part are separated with a non-functional part. That's why it is called split gene. You may write in this way also, when exons and introns are present alternatively with each other, then that is called split gene. When exons and introns are present alternatively or one after another, then that is called split gene. That means when functional part is followed by a non-functional part, that non-functional part is followed by another functional part, like this. When functional part and non-functional part present one after another, then that is condition is called split gene. Then here the exon is the functional part and intron is the non-functional part. That's why as this non-functional part, as this is the non-functional part of the gene, this intron is also called junk DNA, J-U-N-K, junk DNA. This is very important for short question. What is junk DNA? Junk DNA refers to intron. That means the non-functional part of eukaryotic gene is called junk DNA or introns. Okay. So this is the condition of split gene. This is a very simple thing. Split gene. This split gene only found in case of eukaryotic, because prokaryotic DNA consists of only, if this is a prokaryotic DNA, prokaryotic DNA only consists of exons, only functional part. There is no non-functional part present in the prokaryotic DNA. But in eukaryotic DNA, functional and non-functional part both are present. Functional part is called exon, non-functional part is called intron or junk DNA. So when functional part and non-functional part present one after another or alternatively, then that condition is called split gene. Okay. So this much in this class. We will discuss further in the next class. Thank you everyone. Thank you and have a nice day.